So how does Temple assess the differences in these binding affinities? Well, it uses two different scores, QST and FST. QST specifically is the difference between a population using the PWM score, and FST assesses the difference between populations at a specific SNP loci, which is a single nucleotide polymorphism. And then it uses these equations and tells us how the binding affinity for the transcription factor changes, and with the change in binding affinity, thus the change in expression. So what were our results? Now, although we analyzed the X chromosome, the 2L, the 2R, the 3R, and the 3L, we had consistent results across all of them, and so only the X chromosome is pictured, but the trend of the graph is true for all of the FST values that we tested. And so if you look, you have the patterning we would expect to see. So between Africa and California and Africa and Ithaca, we had high diversity. And then between Ithaca and California, that diversity was lowered, which is what we expected to see because of the pattern of Drosophila as it spread out. We saw the same pattern in the QST variation as well. And this is again only pictured for the X chromosome, but we had similar trends in all of our data sets. And so what was our conclusion? In line with our proposed hypothesis, the general trend of the data shows there to be higher divergence between the U.S. populations compared to Africa than between the two U.S. populations. And so going forward, what are we going to do with this information? Well, a large part of our summer was spent constructing the pipeline that allowed us to make these assessments in the data. And so going forward, that pipeline can be reutilized with all of the data sets in the genome nexus. And then furthermore, we went to Flybase and we looked up the genes that had particularly high rates of divergence. And you can see those pictured here. And this is high rates of divergence between Africa and the US. And this is high rates of divergence between the US, Ithaca, and California. And so what you can do with those genes going forward is you can ask why they're highly differentiated between these populations, and then you can use these genes, their human homologs, and their connection to the insulin signaling pathway of which FOXO is a part, and you can use these for further exploration and questions. And so for my acknowledgments and references, these were some of the papers that were vital in our research this summer. And then a special thanks to my mentor, Rita Grace, my lab partner, Kai Cobb, and then to Dr. Wyman and Dr. Hogger here at S8. Thank you.